I've come to Telluride many times. I came here first when I was a little girl. I think I was 12, and I was really excited about skiing the plunge then. I didn't get to do it this past winter, but one day I think I'll do it again. So today I'd like to tell you about some of the work that we're launching in my research group at Colorado State University. And I call my talk Suspended Animation, Exploring Secrets of Cryopreservation. Hopefully you'll come away from this talk knowing a little bit more about what I mean by cryopreservation and be interested in it. If you're curious, these are some images of some cells that have been dosed with some of the materials that we use to cryopreserve them. And this is actually a picture from a facility on the Colorado State University campus, but not part of CSU. It's the National Laboratory for Genetic Resource Preservation, where they preserve all sorts of materials for agriculture in the event that we would have some kind of awful catastrophe. So what is cryopreservation? So there's some organisms that can withstand extreme cold temperatures. So, when it gets down to 0 degrees, we don't really want to go outside without a big jacket and warm clothes on. But these creatures can go down below freezing here. So I've showing here on this slide a few different things. The pine bark beetle. Boo. Yeah. So these guys actually are pretty interesting. This is an adult, and this is the larva. The larva is actually the one that probably is worse in this situation. It can generate a molecule in it called glycerol, which is kind of like ethylene glycol that you have in the your antifreeze in your car. And it helps these critters stay alive at really low temperatures. So they can go down below 0 Fahrenheit to about -4 Fahrenheit, or about -20 degrees Celsius. And in fact, part of their life cycle is helped by having a cold temperature. They actually do better. So they have this situation where having this material in them helps them to be able to stay alive. And then we have the thing, whenever I talk about cryopreservation, well, I shouldn't say whenever I do. Oftentimes when I talk about cryopreservation, the person I'm talking to says something about, oh, do you know about these wood frogs that can go down to freezing and they get all covered with ice crystals? Well, this is one of these Canadian wood frogs here. I don't know if you can see, there is this ice all over this frog. And this frog is alive. Doesn't look like it. It looks like a hockey puck, right? It's frozen. But instead of being frozen and dead, it will warm back up in the spring, and it will use its ability to withstand really cold temperatures. It doesn't have to be at the bottom of a pond to survive in the wintertime, because it generates a lot of glucose in its body. So it makes 100 times more concentrated glucose in its blood than what it normally does when it goes into this frozen state. Now, it would be amazing if we knew how to actually tolerate 100 times more glucose in our blood. If we did that, we would die. So this little guy, when it warms up, it jumps away, and it takes advantage of the fact that it can be frozen outside a pond to breed early in the season and take over the area. So it's to its advantage. And here's this little guy that Judy alluded to. This is one of these, it's a microorganism. So these guys are, you could actually see them with your naked eye. This one requires a microscope to see it. This is an electron micrograph of what's called a tardigrade, or a water bear. And these guys are pretty amazing. This is just a regular micrograph of it, a light microscope picture of it. They exist everywhere. If you go outside to the hillside and you gather some moss and you put it in water, and then you let it sit there for a while, and then you squeeze out the moss, the water from the moss, and you put it on a microscope slide, you should see some of these guys on your microscope slide. But the thing that's really cool about them is that they can go into stasis. They can essentially shut down their entire body and not actually be moving at all, not have even any kind of metabolic activity at all. They can be in a high radiation, high pressure, low pressure. You can find them in space. They go out into outer space and come back, and they can be rejuvenated just by being put back into water again. So people are very curious about these kinds of critters. So there are all sorts of different kinds of things that exist. Fish that live in Antarctic waters, and the list goes on. There are all sorts of different kinds of creatures that withstand extreme cold temperatures. But how do they do it? What is it that they're doing? I've told you a couple of things that they can do, but let's go into this a little bit more. And let's talk about why freezing is bad, right? So all of us have experienced snow and ice, and I selected these pictures of ice specifically because they're really pointy and pokey. And essentially, if you have a cell membrane, it kind of looks like this. There are the molecules that form the skin. Your skin look kind of, they look kind of like these little things. They have little molecular tails that go in there, and they form this what we call a lipid bilayer. And what will happen is if ice can form on the inside of the cell, it can poke a hole in that membrane. And if it pokes a hole in the membrane, the cell dies. Like, that's the definition of apoptosis or apoptosis that you might have heard of, and the cell is going to die. So there is mechanical stress that will happen if a cell freezes, and that could cause the organism to die. The other thing reason that freezing is so dangerous for biological organisms is something that's called osmotic stress. And if you were here last week, I think you heard a bunch about reverse osmosis. This is the same idea here. And that is, if I have a cell, this is my little cartoon, right? If water on the outside here freezes first, then anything that was in the water to begin with gets more concentrated. And the water that's on the inside of the cell goes, I want to equilibrate to this. I don't want the concentration of the stuff outside to be high, so the water leaves the cell, and the cell deflates. And that's actually not so bad. The thing that's really bad is that when the cell warms up again, then the water rushes back in, and that can burst the cell as well. So there are really two different things that are bad about freezing for cells that can kill them. And of course, we're all made out of cells. Anything that's living is made from cells. Well, yeah, is made from cells. Okay, so how does cryopreservation work? Well, I took a picture from a colleague at the University of Utah. This comes from Valeria Molinero's work. She's doing a lot of really interesting things about what are called antifreeze proteins. So cryopreservation has sort of two main areas. There's natural cryopreservation. So that would be our Canadian wood frog and pine bark beetles. And natural cryopreservation can happen through antifreeze proteins that can bind on ice. So you have this is sort of a picture, a cartoon of a protein, and it will bind to the surface of ice. And actually, there also are proteins that are called ice nucleating proteins. And anybody who lives in a ski area should be happy that these things exist, because that's how you make artificial snow work really well. They actually take bacteria, they kill them. They're not alive in the SnowMax packages that they sell, and they spray out when it's cold outside. But that's how they are using it. They're using a molecule, a protein, that's going to nucleate the ice and make ice form. Antifreeze proteins work similarly, and what they do is they bind onto ice, and they just keep big crystals from forming. So if they can keep the crystal from poking through that membrane, then they've done their job. There also are antifreeze molecules. So I talked about glycerol, that's one of them. And sugars are also very oftentimes involved in natural cryopreservation. The thing that we're interested in my research is in artificial cryopreservation. That is the kind of thing that, so I don't know how many of you have thought about, whether or not you can take a person and, totally freeze their body, right? And there's a myth that Walt Disney, after he died, had his body cryopreserved. That's actually not true. He was cremated, but there are other people who probably have had this done. But that is probably not going to happen. I mean, it's certainly not going to happen in my lifetime, and that you can cryopreserve an entire human being. But there are other things that we can cryopreserve. In artificial cryopreservation, what we do is we take a page from nature, right? We might as well use what nature knows how to do. And we find antifreeze molecules that can keep water from freezing. They can change the way that water interacts at low temperatures. And these will include things like sugars, glucose, sucrose. This molecule, trehalose, has a really big reputation of being a really good cryopreservation agent. It's not totally obvious to me why that is the case, but glucose is pretty darn good. And then we have other molecules that we use, glycerol. So you might find glycerol even in some kind of like lotions, you'll find glycerol in them, and ethylene glycol is of course in our antifreeze, and DMSO, dimethyl sulfoxide, is another molecule that we use that can help to keep water from freezing as the temperature lowers. So we're going to mimic nature to protect cells, and here are some pictures of water molecules. Water molecules are, of course, important for biology. We wouldn't be here if there wasn't water, but water is also the offending agent here, because when water crystallizes, when water turns to ice at atmospheric pressures, it generally forms hexagonal ice and has this kind of a structure, and it takes up more space than liquid water does. But liquid water and ice, they both have something that is really important that makes them have the properties they do, and that is something called a hydrogen bond. So what you see is these little red dots are oxygen, and little white ears on here, those are hydrogen atoms. And the hydrogen will form essentially a bond between an oxygen, and those things will hold on to each other really tightly. And in fact, probably you've all seen a picture of something like this. It's a water skater bug, right? How does it sit on top of the surface of water? It's because water is hydrogen bonding. It's holding on to itself really hard. Well, those hydrogen bonds are great for us. They do a lot of things for us, but they also can be problematic if you want to keep things from freezing. So the molecules that we use to be able to help protect the cells all have interactions through hydrogen bonding. So here are some pictures of molecules. Probably some of you don't know our lingo here, but essentially this guy here is glucose, and what I've indicated here is that glucose has these little oxygens and hydrogens, and they can form this little red bond here that is holding onto a water molecule. So glucose is really good at disrupting that hydrogen bonding structure in water, because it can form the same kinds of bonds with water that water forms with itself. Here's a picture of glycerol, and glycerol is actually looks a lot like glucose in a sense. And in fact, if you take glycerol and you can just overlay it onto the glucose, you can see that it's pretty much the same. It's like a piece of a glucose molecule there. And DMSO is a pretty interesting molecule because it can lower the freezing point of water enormously, down to minus 126 degrees Fahrenheit. So it's a really, really low temperature that it can achieve there without letting water freeze. So what we're interested in doing is actually following these molecules like dimethyl sulfoxide, DMSO, or glycerol, and seeing what happens with them, because this is how we actually do artificial cryopreservation of cells here. So what I'm showing you is, I'll show you a little movie here. These are sweet potato cells that have been cultured in solution. So like you would never find, it's not like I took them out of a sweet potato. But before my little movie is going to start here, these cells have already been, the surrounding solution here already has DMSO in it. And what happens to them is you can maybe see here that this little black line here, the cell, what it's called a protoplast, I have been educated by my biology friends. The protoplast is pulled away from the cell wall there, and it's smaller. So you can see that it's kind of smaller here. And when the movie goes, what's going to happen is that the cell is going to, it's going to swell again and get bigger as DMSO gets inside. So essentially, here's our little movie, and what you want to do is watch these guys here, right? And now they're getting bigger. And the people who took this movie, my colleague Gail Volk, said, well, it's because the DMSO that's in the outside here now went inside the cell. But the only evidence for that is the fact that the protoplast swelled again. So we don't really know what went in there, and we don't know really how fast it went in there. We don't know whether or not anything else was important in terms of that. So what we're trying to do in my research is to actually be able to watch those molecules go inside the cell as it's, and then also watch them come out again. Because if you want to preserve biological material, you want to make sure that not only did the stuff go into the cell, but that the stuff came back out again, right? I wouldn't want to put red blood cells in you that still had DMSO inside, because DMSO is kind of bad for you, and you don't really want that inside you. Okay, so this is sort of about what we're trying to do here, and that is we want to be able to watch this little molecule as it goes inside the cell, visualizing it directly. Now, lots of times when scientists are looking to see how things move and where they are in a cell, they attach a big molecule to it that is fluorescent, that lights up. When I shine light on it, it lights up and I can see it there. But if I want to be able to measure how long it takes this little tiny molecule to go across that membrane, it would be like trying to measure how fast Mark can run if I attach a 500-pound ball to him and let him try to run. You're not going to get a very accurate measure of how fast he can run. So if I attach a big molecule to this, I can't do it. And instead what we're doing is we're going to follow the way that the molecule wiggles and shakes, the way that the atoms move in there, and measure how fast these materials can get inside the cell, and then how fast they can get out. And one of the things that's really important about this work is not just about a cell. We know quite a bit about a cell, and in fact, I just showed you a movie where you saw cells where the DMSO apparently got inside the cell, right? But imagine that you have, instead of a cell, you have a kidney, and you want your cryopreservation agent to get inside the whole kidney enough so that the cells on the inside, when I take it down to low temperature, aren't going to die, but not so much so that I kill all the cells on the periphery of that kidney. Because if we could do that, then we could preserve organs for a lot longer than we can do right now. You know, if I dropped dead right now and somebody wanted to harvest all my organs, they'd have to rush to get them to someplace where those people could get my organs to be able to live further. But doing that, they would have to get them to those other people within a day at least. Otherwise, as soon as I die, my organs start to die and the cells start to die. So if we could preserve them even for a few days longer by getting these cryoprotectants in there, that would be great. So why should we care? Judy said a little bit about why we should care. Medicine, anybody who has done anything that's involved in vitro fertilization has undoubtedly had cells frozen, because you're not going to go in there and harvest 8 oocytes and then fertilize them and then implant them all right away. You're going to they always end up cryopreserving them for a while. Tissue preservation is a really, really important thing. If we can actually get a handle on tissue preservation, we'll be able to do so much more to get toward organ transplants and blood preservation. This is a big one that the military is interested in. You can imagine, okay, so this is a related thing. If you could dry your red blood cells out, and you could carry them along with you in a little bag and then just add serum and you'd have blood again. That would be fabulous. So from a different perspective, in agriculture, we also have this. We really want to be able to conserve vulnerable crops. And okay, it's easy to store things like wheat and apple seeds because they're used to going down to low temperatures, but you can't preserve papaya seeds, for example, at low temperatures because those seeds are cold hardy. So you have to find different ways of doing it. So like this is a little picture of a citrus plant that was grown from the shoot that they froze down at liquid nitrogen temperatures and then warmed back up again, and it's growing again. We also really would like to be able to find ways of preserving and protecting endangered species. So if I have a white rhino in a zoo in Colorado, I don't think there's a white rhino in Colorado, and there's a white rhino in Berlin, it would be really great to be able to know how to freeze the sperm or oocytes of those animals. But just because we can actually freeze sperm for bulls, which we do a lot, and in fact the reason that cattle work so well is because they actually breed bulls. That's one of the features that they look for in a bull. They want their semen to be able to be frozen in order to be able to do artificial insemination. And then pest control, right? So those pine bark beetles, it would be really great if we could use their mechanism of generating glycerol in their bodies against them. And then we go even further to think about things that are freeze drying and things like that. So that gives you a little picture of what we consider to be the challenges of cryopreservation. And here are just a few things that my buddies here from Telluride. And I really thank you for your attention and TSRC for giving me this opportunity.